There we go. Hello. Firstly, can you hear me? This is going to be the big question because I've not tried setting this up in the lab before. So this is going to be a bit of an experiment. Can you, there we go. People can see me. Hello, you can hear me. Yes. That means I got it working first time. That's pretty amazing. So hello, welcome to the lab. This is, well, one of the rooms of the lab. I decided to use this room partly because this microscope has the DSLR camera mount to it. So you can see down here, this is what I can see under the microscope. And partly because there's no one else in this room, so I'm not going to be disturbing anyone. Let me just... Choppy video, but audio is good. Okay, well, the video is going to be choppy then. Yeah, to be honest, it's because I'm trying to use the Wi-Fi and that's never going to be great. But what can you do, eh? You do like one frame a second. Okay, so you've got a choice. You can either have more frames a second, but lower resolution. I can drop it down to like 640p. Or you can have it higher resolution, but lower frames per second. Which one would you prefer? Before we get started, because it always takes me about 10 minutes to get started on these streams anyway, because of technical difficulties. Checking the stream. Seems to be fixing itself a little. It's probably because I've got loads of things going on. So yeah, let me know. I don't know how long the time lag is for the comments. Lower res, higher FPS. Okay, that means I'm going to put this on pause and resume. Chat amongst yourselves. And we're back, I think. Oh, that's going up. How's this? I've put you down to, I think, 540p, 480p. I can't quite remember what I put you down to. Yeah, hopefully that's smoother, it still looks good, that's good, and that's what you might be able to see. A microscope's always going to be choppy. Because, there we go, people are saying it's better. There we go, technology, eh? Right, so much going on. So, you join me collecting virgins, and I have had many accusations that I am dealing in the occult, that I'm about to perform some human sacrifices, but instead I am collecting fly virgins. And why do I need to collect virgins? Well, because the work that I do is on relatedness. This is a really bad angle because I need to squint. Hang on. Let's see if I can do this without completely destroying my setup. That's better. So yeah, I need to collect virgins because I study relatedness. And to study relatedness, you need to be able to manipulate relatedness. And to be able to manipulate relatedness, you need to know who is related to whom. So you need to know the exact paternity of all of your flies, because maternity is obviously easy, it's which female is laying the eggs. But in order to know the paternity, you need to make sure that the female has only mated with one male. And the way you make sure that she's only mated with one male is to collect her when she's a virgin and then only expose her to one male. So that is why I am doing it. Is there anyone else in the lab? Yes, but they're in the big lab next door. I am in this little offshoot lab. We have this because this microscope also does GFP, green fluorescence protein, so it can do fluorescence microscopy, and for that you want to be able to switch the lights off. So strange enough, if other people are working in the lab with the lights on, you don't want to be turning the lights on and off. Okay, what is this? Step one, this is called a pooter. It is essentially, it's a long straw, but importantly, how it differs from a straw is it's got a little filter in the end of it. Actually, I'll probably put it under the microscope. Look at this, you're going to love this. Pow! Look at that, it's changed screen. So you can see under the mic, so this is the sucky end. There is a hole in the end of here. Ignore my nails. And this is a tube. This is just a piece of mesh, like a bit of fabric. So air can get through, but flies can't. And then this end is just the bit that I breathe into. So we call it a pooter because you poop with it. Actually, I don't know why it's called a pooter, but you do, you poop with it. So this bit goes in my mouth, this bit goes with the flies, and I can suck them up using this. So it means that I can suck up individual flies in here and then move them into different places. So that's the pooter. Next most important piece of kit is the paintbrush. I literally have my own favourite paintbrush. I marked it with a little bit of red tape because everyone has their own preferences, but the majority of fly work involves pushing anaesthetized flies that can't flap around, with a paintbrush. So everyone has their own favourite paintbrush, and this one is mine. Next we have these, which are vials, V-I-A-L-S. They're not vials, they're actually quite nice. So this is what we keep the flies in, and this brown stuff down here is fly food, and what the flies eat is a mixture of yeast, sugar, molasses, and agar and water. And so basically it's yeast jelly, and the flies, I'm dealing with fruit flies, they eat the yeast and the fungus on rotting fruits, hence fruit fly. And so this yeast is like a decaying fruit. And so we keep them in this. Luckily, we don't have to make our own fly food. We are very lucky in that there are enough fly labs in different departments around the university. Biochemistry works with flies, physiology and genetics works with flies, zoology works with flies. I think there's a couple in biochemistry actually. That someone else's job in the university pretty much is just to make fly food. So we put in our orders at the end of the week, and at the start of the next week, they're like, hey, do you want to come and collect your food? And so we have entire fridges full of this. And in this, we have this yeast. This is just normal baker's yeast, like literally it is for baking. A lot of the stuff that we use is for baking, but it's just dried yeast, and this is more nutritious than this cooked yeast. And so if we want to give the flies a bit of a boost, a bit of a helping hand to help them develop nice and quickly, we give them this, which is what we call live yeast, because yeast, Saccharomyces cerevisiae, I think, is baker's yeast, is a living thing. It's a cell. Is it an animal? No, it's a fungus. It's a living organism. This is it in its dried form. This is the stuff that you put into bread, and you see the chemical reaction is because the yeast is living and is fermenting the sugars. This, however, has been cooked, so this is dead. So sometimes we will sprinkle a little bit of yeast in here, and we call that adding live yeast. Yes, I am at Oxford, for those who are asking. How is it made? You literally just, there's a recipe, it's called Lewis medium. You stick it all into a big saucepan and you mix it up and you cook it and then you pour it in. Sometimes I do it myself when I need to make a special recipe that's different to the standard recipe. It's very boring. Then we have cotton balls. Cotton balls, this is all basic Drosophila stuff at the moment. This has nothing to do with virgins particularly. They just go in the end of the vial, stop the flies from getting out. So this is your standard thing, and then the pooter allows you to take flies from one place, put them into there, put the flies in, done. So that's kind of your standard way of keeping flies. If you want to have lots of flies though, you have bottles. Someone says they totally watch Cooking with Sally. I have made a YouTube video about the science of baking Science, so Great British Bake Off I might have called it, and many of you have been saying, have I seen you on some advert talking about like halloumi or chicken tikka masala or chocolate bar or something like that? Yes, you have. I did a series of science and food videos on another channel as well, which are now going around as adverts. So these are bottles. You can see it's the same food in the bottom of the bottle as it is in the vial. There's just more of it, so you can put more flies in here. These are my virgin flies in here, so I have all of these, and I can actually explain the labelling system to you. So I've got a bottle, and it says WT, that means wild type, which means that it doesn't have any special mutations. Obviously there are going to be mutations because no two flies are identical, but sometimes you get, so these flies normally have red eyes. Sometimes we will breed them to have white eyes, sometimes sparkling eyes, sometimes they can't produce sperm, sometimes they can't tell that they have had sex because that changes their behaviour. So we've got lots and lots of mutations. How the glass chart is plastic actually, but yeah, we had a lab manager who labelled everything. And so yeah, so we've got these wild type flies, which are kind of the most similar to wild flies, as the name suggests. The type of mutation is wild. They're not wild though, they've been in the lab for 40 to 50 years. I think they were collected in the 1970s. So someone in the 1970s went to a place called Dahomey, which I think is in West Africa, went outside, stuck out some rotting fruit, collected a load of flies, and from that we have the Dahomey population of flies, which is in the lab. Other people have done that, different things. And then our flies are kept in huge cages. I say huge, they're only this big, but they have thousands of flies in each one, because there's 150 flies in one of these bottles, just so you get an idea of numbers. And then those flies have been kept in the lab for so long that, yes, they will no longer be that similar to wild flies, and this is always a problem that you have when you're working with lab flies, is how similar is it to wild flies? Can we take the findings from flies in the lab and apply them to wild flies? Well, only with a pinch of salt, but still they're very useful to have, and we've had them for a long time. Are they all dead? No. Okay, so yeah, so we've got wild type, the 12th of January, that is the date when I so-called squirted the eggs. So we collect eggs by getting what's called a pert. There's lots of terminology, but it's not that complicated. It's an agar plate, so instead of having yeast and agar, we make it with grape juice and agar, and make it a bit harder, so that the grape juice smells nice to the females, so they lay all of their eggs on the plate, but it's a harder agar, so the larvae can't burrow into it. We wash off the eggs into a solution, so that we've just got basically just eggs. And then we pipette them. So I pipetted 8 microliters of, as you can see on the label there, I pipetted 8 microliters of eggs into this bottle. And then I waited 11 days, well, 11 and a half days. And during that time, all the larvae hatched and were feeding on this medium here. And then they started to pupate. And you can see that I've got a few different types. Have I got... Yeah, I have. So I've got, I can put it under the microscope. Let me just get in focus. Okay, so you can see this, let's see the other end. That is a pupa that hasn't hatched yet, and you can see the fly inside of it. You can see it's got red eyes. So you can see it's got red eyes there. Just bump up the brightness. So the bright red eyes, actually when you squish a fly, the red that you see is rarely the blood, because flies don't really have true blood like we do. It's probably pigmentation from the eyes. You've got the black there, which is the wings that are developing, and then the rest of this is the pupa. This used to be a pupa, but it has emerged. So the flies come out just like a butterfly coming out of a cocoon. It's exactly the same, except the cocoon in flies we call a pupa, or a chrysalis you might call it in butterflies, and then it emerges, and that is the remains of the larval mouth parts that kind of get left behind. So I should put that lower down the screen for you. I need to raise my chair. And then somewhere in here, let's see, here maybe? There you can see a pupa that hasn't quite darkened yet. And so if any of you are following my Instagram on Sunday, you'll know that I posted a photo saying, oh no, my flies aren't ready yet. And that's because the fly pupae were all white, and they turn white. It takes them about 5 days as pupae, maybe no, 4 days as pupae. It depends on the temperature. And the first 2 days they're pale, then the last 2 days their wings darken, and that's how you know that they're about to emerge. So my flies weren't dark when they were supposed to. It's because they've been running about ½ a degree colder than they normally are. But now they have emerged. So how do you collect virgins? Well, this morning I came into the lab and I got rid of any adults that had already emerged because I don't know about them. And this is a bottle that you can see is kind of gunky, so I won't be using this for collecting virgins. But you can see under the microscope, you don't need under the microscope, it's just a bit clearer than the webcam. You can see that there are flies running around in here. And so these are flies, actually just a quick, I can show you how easy it is to sex a fly. I mean, we'll be doing sex and flies in a bit, but okay, so we've got that's a male, it's a female, 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 male, male, female, female, male, 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 male that only just emerged, female. So yeah, you can tell male, female quite easily. So what you do is you grab, let me switch over to, is it still working? Is you grab the morgue, which is literally called the morgue. It is a bottle of 70% ethanol, the rest is water, and this is how we kill flies. You have 2 ways of killing flies. If you don't care about them, you stick them in the morgue. If you do want them, as in you want to count them, for example, or look at them after they've died, then you'll put them in the freezer. So what you do is you take a bottle and you dispose of the flies, and this is how you dispose of flies. So these are ones I don't care about. So tapping down means that all the flies go down to the floor, and then they all start to crawl up. You can see why I don't normally use this bottle because all the food's now come loose. And there you go. Now I've cleared anything that was an adult. You can kind of see what it looks like in there. I mean, this is a really messy bottle, which is why I'm not using it. But you can see there are no adults flying around anymore. So all I have in here are larvae and pupae, no adults at all. And this is how you collect virgins, is knowing this one bit of biology about flies, which is that it takes, at 25 degrees C, it takes an adult 8 hours from emerging from its pupa, what we call eclosion, that's just the stage of emerging as an adult, to becoming sexually mature. In those 8 hours, it's still pumping itself full of fluid, kind of stretching out its wings, getting used to being in the adult stage, not in the pupal stage, feeding. So in that stage, it can't have sex, basically. So what you do is you get rid of all the adults, because they're too old. Then you wait for about 6.5 hours, and then any adults that you have that have emerged within those 6.5 hours, you know are too young to have had sex yet. And so all of these are virgins, because I got rid of anything that would be old enough to have basically, and as well, if they're old enough to have sex, they will have had sex. Flies have sex really, really quickly. So that's how you collect virgins. Someone said they don't have pronounced larvae and pupae. So a singular larva, one larva, two larvae, one pupa, two pupae. Okay, so no, adolescence I would say is the larval stage. Puberty is probably the pupal stage. This is as an adult, as a young adult, it takes them a while to emerge. So now we've got these virgins. How do we keep them as virgins? Because obviously, within the next 2 hours, they're gonna sexually mature and start having sex. So I need to stop them from having sex. And the way you stop them from having sex is to separate out the males and the females. And if you keep males separate from females, obviously they can't have sex, and so I can keep them as virgins for as long as I want. And that means that I need to sex these flies, which is why the alternative title for this video is How to Sex a Fly. And you can see that I can do it visually because I was doing it before. And I need to take all of the flies out of here, sex them, and then put them into single-sex vials like I showed you before, some for males, some for females. And to do that, I don't want to do this while they're all flying around. So what I could use is, you can't see it, it's just off camera, but there's a CO2 tank here. The CO2 replaces the oxygen in the air, displaces some of the oxygen, so they kind of go into, they can't move around because they've run out of oxygen for a bit, and that knocks them out. But that's not great for them. And as virgins, particularly when they're pumping themselves up, you don't want to really anaesthetize them with CO2. So instead, we rely on the fact that flies are cold-blooded. They are ectothermic. They rely on an external source of heat. And as you cool them down, their activity slows down, which is why we have a box of ice. We get through a lot of ice in the lab. And I have this empty bottle cooling down on the ice, and I have also got a petri dish here that I filled with ice at the start. That is cooling the petri dish down. So I need to, let's move this to one side. I need to take my adults and I need to anaesthetize them on ice. So this is what I do. I don't have to stand up. I don't have to stand up, but I want to stand up. So I take a bottle, and like I said before, when you knock flies, when you disrupt them, they fall down. That gives me time to open the lid and tip it upside down before they all fly out. You'll get 1 or 2 flying out, but we've got fly paper stuck around the lab. It's not too big of an issue if we lose a few. Yeah, they will just chill out once we put them on ice, exactly that. And then we'll tip them down. They'll hit this cold surface, and also because it tapers up and there's a funnel in here, it's quite hard for them to fly out. So eventually they'll hit on a cold surface and they'll be like, oh, it's so cold, oh, I can't move anymore, and they'll just stop moving, but they're completely alive. Yeah, so this is how we anaesthetize them, and it looks like this. There's a lot of tapping involved in fly lab stuff. And I don't even need to worry about collecting all of them this time because I've got so many of these bottles. Yeah, just soften the melanogaster on the box. So now if I was to leave these bottles for another 6 hours, then any flies that were adults would be virgins, because I've cleared all the ones that have already emerged. Now, I don't actually need all of these, but I might as well collect them all. They're only going to die anyway, so might as well. I don't want them to remain virgins forever. I will be mating them, but I want to make them under controlled circumstances. And then after I've mated them, I don't care about them, so then I'll kill them. So yeah, people have been talking about virgin sacrifice. If anything, the virgins stay alive longer, because anything that's not a virgin gets killed. And at least the virgins I keep for a few days to use in my experiment, and then I kill them. But yeah, so if you're in a fly, being a virgin means that you're more likely to stay alive for a bit longer. How long will I be streaming for? It's hard to tell. This process would normally start to finish take me about 20 minutes. But obviously I'm explaining every step to you. So, I don't know, maybe another 40 minutes? I really don't need this many bottles, but hey. How many are going in the ice container? Thousands. I can show you after they've been knocked out. About the title, I am collecting virgins. Like, literally, that is what I am doing. I am collecting virgin flies. It's great because you have to come in the morning and the evening. Often I'll be like, oh, sorry, I've got to leave the party. I've got to go harvest some virgins. Because that's the fun stuff that fly scientists do. The most athletic flies they can get out, yeah, the most athletic ones. Very few escape, but some do. But we do have, I mean, the door is sealed, there is netting the other side of the door, there is fly paper up in the lab, it's ventilated as well, so it's going to be, so the flies won't make it outside, but they might survive in the lab. So you can see there's actually a few, you can't see, there's a few in here. So, these are pretty much cooled down now. So you can see in the bottom that I've got all those just anesthetized flies. Okay, so whilst they're cooling, because I don't need these bottles anymore. Because if I was to take them out now, yes, they are anesthetized, but they'll warm up really quickly from the stage they're in now, so I want to keep them on for about 5 minutes or so. I'll also replace the ice in here, because you can probably see it's melted. This will be useful later on. This is how we keep them on ice. This is like the champagne bucket equivalent for flies, is a petri dish filled with ice. Like I say, there's a lot of tapping in the lab. So this is just a petri dish with a filter paper stuck to the top to make it white. So we'll come back to that later. Now I'm going to start labeling my files, because once I have sorted my flies into male and female, then I want to be able to keep them in male and female things. Someone's asking about security things. They're strictly speaking genetically modified. Well, not these ones in particular, but some of them are. So strictly speaking, we're not allowed to release anything genetically modified, just because that's the law. However, our genetically modified flies are shit flies. They are genetically modified to be worse than normal flies. So it actually won't make any kind of ecological difference if they escape, but that's wise. Because like legally, if I was, I don't know, making genetically modified weeds that are super resistant to weed killer or something, you don't want it to escape. But there's no real reason. They're not in any way harmful. Just wanted to. Okay. So I'm going to put some live yeast, like I explained at the start. Worse in what sense? Worse in that they are not as good at producing offspring. So they are less fit in an ecological sense. They will produce fewer offspring than wild-type flies, and so there is no way that they will be able to outcompete other flies. So all I'm doing here is taking a normal vial and sprinkling a dusting of yeast. So you can see you've got the live yeast grains in there. Actually, I can probably put it under here. Yeah, there we go. You can see that there is yeast in there. I'm not assuming gender. Flies don't have gender. Gender is a social thing. Flies do have sex. They have males and females. Males and females refers to sex, not to gender. So you can't misgender a fly because gender is a human thing. Flies, I don't know if you know this, guys, but flies are not human, and therefore flies do not have gender. I mean, sometimes you can get some weird sex mutations. So I know one lab has made flies, female flies that chase males as if, female flies that chase females as if they were males. Which is quite an interesting thing, kind of masculinizing the brain. But of course, remember, these are flies, so don't go applying any of this to humans. Okay, so now I have so-called yeasted the vials. I know, right? Worldview is shattered. Flies are not humans. Okay, so yeah, my cotton balls. Normally we have huge bags of this, but because I'm working in the small lab, I've just got these. It's going to plug the ends up. It'll make it easier for me later. Remember that I've got my virgins on the ice just down here, which you can't actually see because I should move this over here. Look at that. That's going to be more sensible, isn't it? That's much more sensible. Ta-da! So, yeah. In this bottle on ice are my virgins. I'm just letting them chill for a bit. There's no rush. As long as I get to them soonish, it's going to be okay. Oops. I'm not even doing this quickly, like seriously. The setup time involved just in getting this camera set up was a lot longer than it would normally take me to do the entire thing. But I don't have anything else to do today except weigh out a quantity of yeast and add a fixed quantity of water to it, ready for tomorrow. Woohoo! So my experiment is actually going to be starting. Day 1 is on Friday, so today is day -2 because I don't include zeros because it's 2 days before the start of my experiment. That is because, so you know how I said that adults take 8 hours to reach sexual maturity? Well, they then, once they become sexually mature, i.e. if they have sex, they can produce offspring, it takes them a while for their production to ramp up a bit. So they can produce a few, I say a few, maybe 50 eggs a day on their first day, 70 eggs a day on their second day, and then up to about 100, 120 eggs a day on their third day. And then as they age, it goes down again. So older flies, one of the signs of aging that we use is reproductive aging, as in my last paper, which is open access, so you can read it if you want. They produce fewer eggs a day, or if they're males, then they are less successful at siring offspring each day. And so I want to get these flies to lay as many eggs in as short a period of time as possible, so I'm collecting them now in order to make them on Friday. And I'll just keep them in these vials for 2 days, just to let them age. Seems like a lot of this work could be automated. Yeah, but the trouble with automation is that it's fine if you're doing the same thing over and over again. But often we're doing the same thing but ever so slightly differently each time. And also automating things costs an awful lot of money. And so to be honest, it doesn't make financial sense or scientific sense. So if I was working in industry and literally all I did was the same task over and over again without ever having any changes, it would make sense to automate it. But every experiment is slightly different. Because it's science, I'm always testing something slightly new. Every member of the lab is testing a slightly different thing, so it doesn't really make sense. So for example, we kind of automate, we don't automate, but we have division of labour when it comes to making the vials of food, for example. We don't make our own food, biochemistry makes our food for us. And it's really not that time consuming. And I'm the sort of person that writes down the exact number of hours that every task takes, because often I'm working in multiples of 120. So if something takes twice, let's say something takes 1 minute, but someone else takes 2 minutes to do the same job, that's an extra 2 hours. So things, small per unit time changes can make big overall differences. So I'm very obsessive about writing down how long everything takes. But yeah, it's not such a... Have I done any experiments involving other creatures? I did my undergraduate project on ladybirds. I was looking at the harlequin ladybird, which is an invasive species in the UK. It's also an invasive species everywhere except Siberia. America deliberately released it. They were one of the first people to release ladybirds, Yukon ladybugs, as a biocontrol agent to reduce aphid numbers on wheat. And then the harlequin is so good at being a ladybird that it beat all the other ladybird species and kind of wreaked havoc on native ladybird species. And fair enough, America didn't know, no one had done it before. France then stupidly, after seeing that America had had a terrible time introducing the invasive species, decided to also introduce harlequin ladybirds, and they then flew across the channel and we've got them. So I was studying that. But those are the only animals that I've worked with. Okay, so we've got our flies here. I'm just going to label male and female. So you may be familiar with that sign as the sign for female. If you stick a little hat on it, that is the sign for virgin female. You can't really see that. There we go. That is the sign for a virgin female. Males, I mean, you could write a little V, but you don't care about the virginity of the males. You only really care about the virginity of the females. Read into that what you want. And the date today is the 23rd, so I'll just label. I'll label a few of them, and you'll see why in a sec. And yes, this is an actual experiment that I'm doing that will be written up into a paper. So I've got 4 of each. Okay, so now, some more paper. The problem with using ice rather than CO2 is that ice is messy, ice melts, you get water everywhere, it's a bit of a faff. So I tend to stick everything on paper towel and just kind of reduce the puddles. Bring the microscope over. Kind of focus right. There we go. So the things you can change on a microscope, if I switch over to this. So I can control the amount of light that comes into the microscope. I can control how far apart my eyes are, that's just for comfort. And if I have a difference in the prescription of my eyes, you can change that here as well. I can change how zoomed in I am, which on this can go from 6 times magnification to 0.75 times magnification, plus maybe a bit up here. And then I can change the focus as well. Okay, so I am going to do a small amount first. So this is the stage where these are virgins, they've been anesthetized, they're on ice, they're happy, they're fine. It's like they're in a kind of hibernation. So I'm going to tip a few out of the bottle because I want to be able to show you and talk about them without worrying about them waking up. GFP, I do sometimes use GFP. At the moment I'm not using GFP, and yes, I will switch the main lights off. So prescription is called, it's called diopter. You get that on most binoculars as well. You can just change the individual thing up here, change the diopter. Okay, so what can you see? Right. Why it made the noise, but it did. So these 4 are the ones I want to talk about. Okay, so now you see why we get really pernickety about our paintbrushes, because under the microscope paintbrushes look completely different. This is an adult male. I mean, they're all adults, but that's a mature male, that is a mature female. So now we're on to how to sex a fly. Sorry, just moving it so I can, because I can see more down the microscope than you can on the screen. Okay. So, that's my map. Here we go. This is a male. This is a female. I should have got myself a needle. Normally we have mounted needles if you want to point to things, but I don't actually need a needle for this. I'll try and use the end of paintbrush, but it's a bit big. That black spot is the male genitalia. That is what tells me that it is a male. Just put the light up. So that includes the kind of penis-y type structure, some claspy structures that clasp onto the female during mating, and that is what the male uses to transfer sperm over. And you can see it's kind of this dark splodge, and that is a male. And actually that's melanogaster means black belly, and it's because when I flip him over, actually, you can see that he's got black on his bum, on the top side of him. So that is why they're called melanogaster, because they have this black bit on their bum. And Drosophila, Drosoph means dew, as in the condensation at the start of the day, dew. Phila, loving, so dew-loving, black-bellied flies. Drosophila and melanogaster. Yeah, they are different to humans. This, on the other hand, is a female. And you can see that, firstly, she is bigger than the male. She is a different shape to the male. So she has a much fatter belly than the male has. So you can see she's quite a bit bigger than the male. And if I zoom into her reproductive organs, her bum ends in what I describe to my students as a nice little tip. It's a neat ending. That includes her ovipositor in there. So compared to the male, who's is kind of like, it's almost like his bottom half has just been ripped off. It just ends really messily. The female's comes to a really nice point. So that's the male and the female. And yes, this petri dish has got ice in it to keep them cold, even though they're off the main box of ice. However, if I go to these flies, you can see that these ones look a little bit different. And that's because these have only just emerged. So if I compare these two, these are both males, but the darkness, they haven't, this one's just emerged compared to this one. And so it takes them time to darken, so they're paler, and you can see the shape is a bit weird as well. And so actually I know that this is a lot younger than this fly. So even if I've got a mix of virgins and non-virgins, I would know that this fly was still a virgin, just by looking at it. But you can still see the characteristic black dot just there, which tells me without fail that this is a male. This is a female, and you can see that she looks completely different to this female. These are both females, but this one has only just emerged. You can actually see that her wings haven't expanded yet. So when they're in the pupa, sorry, I'll just put this for you. When they're in the pupa, obviously their wings are all folded up. But then when they emerge, they have to pump them out to straighten them out. And so her wings still haven't been fully pumped out yet. And if I look really closely, I'm not sure I can spot it on this one. Maybe I can spot it on the male. Yeah, so you can just about see, not very clearly. I'll find another one. But you can actually see what's called the meconium. I'll point it out next time I see it. But you know how when a baby has its first poo, and it has this really weird poo because it's the one poo that it's been holding inside of it for like 9 months, and it's really weird, and then it gets rid of this really weird poo, and then it starts having normal outside of the womb poos? Well, flies have this too. All of the excreted waste when they're pupae goes into their this one weird poo called the meconium, which is green, and because their bellies are slightly translucent, you can see it through their belly, and that's another way of telling that they're definitely virgins because they haven't had their first poo yet. So we've got, let me turn the brightness down again for you. How long do I have until I warm them up, basically? So we have normal male, I have freshly emerged male, normal adult female, freshly emerged adult female. And then I'll sort them out. So I've got some here, so I'll put my males on the left and my females on the right, and I can go, so I can just focus. So I can do this pretty quickly now. Just bring them over here. So I've got male, female, male. Actually, it takes me longer to physically move them than it does to sex them. So all these on the left here are males. You can see they've all got that little black splodgy bit on their bum. And all these on the right here are females. You can see they've got no black splodgy bit, and they've got a neat little point. And so I've now separated out my males and females. I've got 1, 2, 3, 4, 5 females, and 1, 2, 3, 4, 5, 6, 7 males. And quite handily, I like to keep my females in groups of 5 and my males in groups of 10 because I want as many eggs as possible. So I want my females to be in tip-top condition, so I don't want to crowd them too much. Males don't care so much about crowding. This is where I get the pooter. And I will suck up the flies and transfer them. So this is my female vial that I labelled before. And I will suck up the females. Where is it here? See that? Yeah. So now they're in this bit. I will put them in the tube. I'm missing a fly. Right, okay. So now they are in here. And as they come up to room temperature, they'll start to come back to life again. In fact, if I put them so there, you can see that she is already moving around. She's a bit lethargic, but she's still moving around. These ones over here, they're still a bit cold. They haven't got their legs up, but so I've got my fingers underneath so that my body heat will warm them up a bit faster than it was. So this is just speeding up the process. But you can see that as they warm up, their legs start twitching, and then eventually they'll right themselves up and start wandering around. Oh, there you go. You can see the meconium in the bottom of that one. You can see she's got green in the bottom of her. That's the poo that I was talking about. And there you go. She's waking up now. And so if I see this when they're supposed to be on ice, I know that I've got not much time before they start moving around. So now they're up and walking. Most of them are. Oh yeah, they're all up and walking. They're still a bit groggy. They'll take a few hours to come back to fully normal, but they're better than they were before. So that's my 5 females in there. I'll get rid of the males. And that's my males in there. You've always got to make sure whilst they're anaesthetised that you don't squish them between the plastic and cotton. Okay, so that is collecting virgins. Now, these are virgin females, these are virgin males, and there the twain shall meet, and as long as I keep them separate, they shall remain virgins. So, I've just got all of these flies to sort through. I don't need all of them, but I need quite a few. So, I'm just going to drain some of the water from this. So now I'll do it at real speed because obviously I've been talking you through. So I will tip out the flies. Probably a few more than I would normally do, but so they're not frozen, they're just cold. They're still liquid because on ice they're only at like 2 degrees maybe. They're not frozen solid. Okay, so what you do, yeah, this is quite a few more than I normally do, but hey, you can get the idea. Just going to spread them out, so 1, I can see them more clearly, and 2, so that they've got better contact with the cold Petri dish. And now you can see why we've got filter paper on top of the Petri dish as well, just so that they've got a white background. Okay, so now let's do females on the left, males on the right. So this is actual speed. And obviously when I'm brushing, I've got to make sure that I don't squash them. And this is why I usually listen to music or podcasts when I'm doing this. Although sometimes I then get confused and forget which side I've put which sex on. So it doesn't matter. I mean, you can see it. So there, you can see the dark spot in the middle of this one. And just in the middle, that's the meconium. That tells me that it hasn't mated. Well, not that it hasn't mated, but it's still young. Oh, I messed up that fly. Whoops, that fly is now a dead fly. Oh well, I've got loads. I mean, this is the thing. You do kill them, and you just got to balance speed, accuracy, and delicacy. I'm usually pretty delicate, but sometimes they just get caught under the brush in the wrong way. And these red smears, you can see, that's where someone has squashed a fly against the white paper, and their red eye pigment has gone everywhere. Another thing you quickly learn when you're doing a lot of microscopy is I have both of my eyes open at the moment. A lot of people, when they start off using a microscope, have real problems with binocular microscopes. And that's why you have to adjust the interocular distance, the distance between the eyepieces, so that you can use both. Another problem is that this is taking up my entire field of view. And I'm not moving, but my field of view is. So when you first start on a microscope, if you're like me and you suffer from travel sickness, you get the worst headaches ever because you're focusing. Often you've got quite bad posture, but also you get travel sick using down a microscope. And when I was doing like 8-10 hour microscope days, I would actually take travel sickness pills, even though I wasn't traveling anywhere, just because the thing that causes travel sickness is a difference between your brain and your body's sense of movement. So your eyes and your body's sense of movement, I should say. So my eyes can see movement because I'm moving the plate around, but my body isn't moving, and that confuses your brain and makes you sick. Which is why I try to, instead of slowly moving the plate around like this, I will push it to the edge and then move, and then keep it still, and then move and keep it still. Yeah, so ginger's supposed to help, but I mean, these things only help to so much when you're doing this a lot, and usually you're quite squat. I'm really squashing a load of flies. I can't talk and push at the same time. When you're doing a lot of it, you don't want to be feeling nauseous, because usually you're tired and stressed out because you're in the middle of an experiment anyway, and that always worsens it. So now you can see I've just worked my way down the plate and I've sorted them into 2 lines, males and females. And that's what, I don't know, 250 flies in 5 minutes. You can do it pretty quickly. Yes, compound, stereo microscope, normal paintbrush. I don't know how the paintbrush would be different. Okay, so now I need to recruit them. And remember, I want 5 females in a vial, 10 males. So, roughly, it doesn't matter too much. I'm going to keep my females to the left of me. And the annoying part about this is if you've got someone on the microscope next to you, it's great, you can chat, but if you've got a pooter stuck in your mouth, you can't talk, or you try and talk and then you end up blowing them everywhere. It's terrible. And now, because I've got enough labelled vials over this side, I'll just go through these and then label them all afterwards. I don't know which bit's the interesting bit. Is the interesting bit me or a pooter? I don't know. Which bit do you want to see more? The pooter or me? Now you can see why I got all of these vials done in advance. And you will get some wrong, so if you miss sex a fly, you've got to remove that vial, which is why I don't put too many in one vial, because if I put a male in here, that's wiped out all of the females in here, because I don't care about them, because they're not virgins. So. Sometimes I get stuck at the end of that, which is a bit annoying. I think I've got a 6 there, but oh well. Yeah, 6. Take them out. research am I doing with the flies? It's in the FAQs under the video. Sorry, I can't really speak right now. But that is a question I get quite a lot. Alright, that's all the females. That's 1, 2, 3, 4, 5, 6, 7, 8, 9. 90 females. That's not bad. I'm going to need 120 for my experiment, so I've collected far too many, but it's fine. So now onto males, and I can do males in groups of 10. Well, it really doesn't matter with the males, roughly 10. To be honest, I can go up to 20. It just saves on space. Dead. Ish. They will be soon. That's exactly 10. You get quite good at eyeballing what a certain number of flies will say. I'm going to take 11 there because I think one was squashed. See what I mean? Without talking when you're doing it, you drop them everywhere. See, now you can see they're starting to wake up. So now I need to act quickly. Still got a few minutes though. Mostly it's because the ice in the petri dish is melting. I need to replace the ice. And you give them names if you want. Now you see why people say, oh, you named your flies. I deal with thousands of flies on a daily basis, so no, I don't name them. Wriggling around a bit. Right, so that's those ones done. Why is that not working? Oh, I need to change the battery. Bear with. Okay, let's put those squashed ones in the morgue. Right. These are not wild caught, these are culture. What am I doing? Replacing this. So yeah, you can see that this is now all melted, which is why they were starting to wake up, because they were no longer cold enough. So I'll just get some more ice. though you've seen how big the flies are, you can hopefully appreciate that this entire black layer is dead flies. We get through a lot of flies in this lab. Luckily, flies are not considered under animal rights by the UK government. Otherwise, that would make our job infinitely harder. We actually had to worry about the well-being of our flies. Oh my god, can you imagine? Do they die in the morgue suffocating? No, they kind of drown instead of suffocating. Yeah, they drown, get tipsy, drown, die, something like that. I'm going to want to make some more files. Always collect more flies than you need, because you never know what's going to happen. Why are you not wearing a white lab coat? Lots of people think that white lab coats are kind of like a uniform for scientists. They're really not. They're just a protective gear. And so one of the hilarious things, like, there's this chemist that's new to Oxford called Kyles, and they wear white lab coats in the shop. It's like, what are you doing? You're not dealing with harmful chemicals. And even if you were, a lab coat should never be worn outside of the lab, because the whole point is that the chemicals go on the lab coat. So you don't want to then have those chemicals on the lab coat going outside of the lab and contaminating what would otherwise be a safe area. So if ever you see a lab coat outside of a lab, think very carefully about what they're doing. So yeah, you only wear a lab coat if you're dealing with something that requires a lab coat, which is usually some kind of harmful chemical. So it's very, very rare that actually I wear a lab coat. I wore one earlier today because I was dealing with Nipogen, which is an antifungal agent, which in its concentrated form is slightly harmful. And mostly because I was dealing with grape juice and I didn't want it to stain my clothes. And because wearing a lab coat makes me feel like a fancy scientist. But 99.9% of the time I don't wear a lab coat, because you only wear a lab coat if you need protection against something, and I don't need protection. Yeah, you don't need a lab coat in physics labs, unless you're dealing with some weird chemicals. Especially in our fly labs. Most of the stuff in the fly labs you could probably eat. You shouldn't, but it probably wouldn't kill you if you ate it. We were having this conversation earlier today of, if you're stuck in a lift, which bit, do you eat the fly food or do you not eat the fly food? It's the kind of conversations we have in the fly lab. Do I have a title? My title is Ms. I'm not a doctor yet. But Gavin wears the lab coat, yeah, because Gavin wants to protect his clothes from paint. So it's essentially overalls rather than a lab coat, which is kind of all a lab coat is. A lab coat is overalls, but for harmful chemicals. But yeah, he doesn't want to get his clothes stained, so he is wearing protective equipment so as to stop his clothes from getting stained. I did not bring enough cotton balls into the lab. And so yeah, the same way that I don't want grape juice to get on my clothes, so I'll wear a lab coat for that. How many fruit flies have I accidentally swallowed? I don't know, not that many. Very, very, very few. And if I do, it's not because I was pooching them, but just because they flew into my mouth when I was talking in the lab. Right, let's get his last tree done. And then these will all go in the bin in the wall. So I can tell that that's a newly emerged one because it's paler than the others. That's a newly emerged male. Newly emerged female. The newly emerged ones are hard to tell apart. That's newly emerged female, newly emerged, the wings are slightly different. Obviously mature males, you can see, have this really prominent dark bit on their bum. And mature females have this really big white underside, whereas when they are younger, they're not quite as distinct from each other. What am I going to do? Males on the right again. Someone's getting hungry. I'm getting hungry. I had a brownie earlier, but I swear the more I eat, the more hungry I get. If only I knew someone who was scientific who could explain that for me. Just wait, so after I've collected all the virgins that I need, I'll be putting all of the excess virgins that I don't need in the morph. So that will be a mass sacrifice of virgins, a mass murder of flies. I put them on the wrong side, you're a female, you should be on the left. Yeah, that will be the finale of the video. It will be a mass killing of several hundred, if not thousands, of flies. Look forward to that. What colour does a fly see? That's a really good question, Marie. I don't know. I haven't looked into fly optics. I don't know which colours they perceive most strongly. Most animals have a bias towards certain colours. So as humans, we have a bias towards red. We're very good at distinguishing red things because red tends to be ripe fruits. And it's why our danger signs, our stop signs are in red, because we're very good at spotting red. Most mammals aren't as good as us at red. Most mammals are colourblind. Birds are very good at red as well because they also eat a lot of fruit. I don't know what, like bees are very good at UV and blues. I don't know what flies are particularly good at seeing. That's an interesting question. I know that the mutated flies, so the ones that we breed to have white eyes, we breed them to have white eyes just so we can recognise them a little bit easier. They're not as good at seeing as the wild type red-eyed flies. So the red is an important thing. The fact that it's red isn't necessarily important, but the thing is important and it so happens to be red. Am I Lord of the Flies? Yes, I am. I am Lord, Ruler, Dominion, God. I mean, it must be really weird for them, right? Like, they end up getting anaesthetized, and then the next time they wake up, they're in a completely different place with completely different flies, so they're in single-sex vials, and they have no idea what's happened in between. And then occasionally, even when they're awake, this weird tube thing comes down from the heavens and abducts them and moves them to a completely different place. Like, it must be really weird. I hope you're playing along with guessing which one's a male and which one's a female. Everything is black? Oh no. Is it really? Can you see me? Can you not see me? It looks fine on my end. Tell me if you can't see me. Stream is fine, okay, good. I don't know what the time limits of the stream is, because at the moment I'm not a YouTube partner because they're being annoying, and it just so happened that I left my MCN at the same time that they changed the rules to become a partner, so they've got this huge queue and I'm waiting in the queue to become a partner. So even though I was a partner before, they haven't re-registered me. And what was I saying? Yeah, I hope you're playing along at home as to which one's male and which one's female. Because we should be able to tell just as well as I can. And done. So those are all my females. Those are all my males. Spread them out a bit. Make them easier to poo. Also helps me spot if I've gone wrong. I've gone wrong. Last view. I think I've got shapes from that one. Yeah. Elusive gamer, am I single? What difference does it make? To you, at least. Makes a difference to me. But this is a boring bit, so if you've got any more questions, let me know now, and then I'll be finishing up in about 10 minutes. Hello, fly, yeah. Everyone has. 28, that means I've got 140 fly females, but I want to be more, because I need at least 120. Now wait, every butterfly is their model organism. No, but other people do, but not me. Can you taste them? Kind of. So because I'm breathing in, I can taste kind of the yeasty smell. And so when you're dealing with different vials, you can kind of taste that. Like if I'm sucking up from the pooter when I've been using CO2, you can taste the CO2, like the bubbles, like when you get bubbles up your nose when you've been drinking a fizzy drink. So I can kind of taste the air rather than the flies themselves. Can we use a microscope to zoom in on the fly eye? Yeah. Can you do it afterwards? Got plenty of flies. You can do it right. Yeah, the ones on the left are female, the ones on the right are male, and they're the females at the moment. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 1, 2, 3, 4, 5. So I've got 50 more than I need. That should be alright. Finish them out. I think I've got a female in that one. No, it's very young now. Got 11 in that one. 11 is close enough for 10. This is just for keeping them, they're not even the experimental flies, so it doesn't matter too much. Can I not do this part by eye? Yes, but it's just as easy to do it down the microscope, because I can see where the end of my creature is. And I can see how many I'm picking up a little bit easier down the microscope as well. Twitch TV and revenge. Flies do not have gender, they are not human, therefore they do not have gender. Therefore there's no such thing as identifying as they're male or they're female or they're some weird sex mutant. quantitative data because the stats is easier and it's more powerful. How many have I got? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So I've got about 170 of each. That's fine. I can't be bothered to sort anymore. Okay, so they're all sorted. I'll save the labeling until I'm off camera because that's going to be really, really boring. Last few things. Someone wanted to zoom in on the eye of one of these flies. Interestingly, actually, you can see there's a little black spot in the centre of each eye. That's called the fovea. It's just an artifact of how the light bounces off their multi-eye. So as I was to move the fly around, that black spot would change. That's as far zoomed in as I can go. I'll make myself smaller. Let me just focus. So there you can see the individual lenses of the compound eye. In between each lens, there's actually a tiny little hair that you can't quite see, but you can then mutate the shape of those hairs, and that gives the eye a slightly different appearance. So that's one of the ways you can tell the difference between them. Another mutant you can get is you see these hairs on the back of the thorax. So a fly is an insect, so it's got a head, thorax, and an abdomen. These hairs are long and come to a very fine point, whereas you can have some that are called stubble, where it looks like you've shaved the fly because the hair comes to a very blunt end and it's shorter. So that's another mutant that we use to identify flies. So for example, we might we might have one parent is normal hairs and one parent is stubble, and then by looking at the hairs on the offspring, you can tell who was the father. Flies are in the order Diptera. Ptera means wing, so pterodactyl, wing finger, Lepidoptera, scale wing. What other ptera things are there? Pterosaur, winged lizard, Hymenoptera, membrane wing, Diptera, two wing, because they've got just one pair of main, where's my paintbrush gone? Here's my paintbrush. One pair of main wings, these are the main wings, and then that little, I really need a needle, that little blobby thing next to the, behind the leg is, oh, sorry, is the haltea. And that control, that's kind of like the stabilization system of the fly when it's flying. It uses that to make fine scale adjustments to how it flies. What other things can I show you on the flies? Show you the mouth. You're waking up. So you can see the mouth on that one there. They taste with their feet. They've got little bristly antennae. You can create weird ones out. Yeah, this is what it looks like. I don't know what else you'd want to see down the microscope, really. There's not that much else you can see. So the females. We can see the wings nice on this one. I love the female wings, they iridesce so beautifully. I don't know why, but I think I find the female wings are prettier. And you can see that the wings have tiny, tiny little hairs at the end of them. The Does anyone take home extra flies for pet food? Funny you should say that. There's a group that studies spiders in the department, and they sometimes come in and say, "Hi, can I have some flies to feed our spiders?" Strict, because you're not allowed to take them out of the department, because they're GM. It's really not a big deal. So you can see that they look pretty similar. There's this profile view of the male genitalia. Have I done much study on the wings iridescence? No, I have personally done zero. Knowing the paternity of the offspring allows me to study the flies. How do I study their behaviour? By watching them. Switch over. You literally just watch them. So you can watch them mating, you can watch them fighting, you can watch them feeding, you can watch them courting. Yeah, you just have these vials up on the rack and you literally just look and you're like, oh, okay, so that's what those ones are doing, that's what those ones are doing. When did I know this is the career that I wanted? This isn't the career that I want. The career that I want is YouTubing, not this. Let's put this back there. How many hours do I study a day? Not enough. It really depends. Experiments, you can be working 12, 13 hours a day, and then when you're not doing experiment, you want to sleep, so you don't. It changes. Mass cull time. Thank you to the 116 people for watching 1 hour and 40 minutes of collecting virgins. I have all of these virgins in here that you can see. Those are all virgin flies that I do not need anymore. So let's do a virgin sacrifice. I don't know if you want to do some kind of weird chant or something. I don't know. But here is the morgue. So here are the flies that I'm going to brush them into the morgue. Try not to squish them so that they don't make a mess on the petri dish. And then, I mean, I can pour them closer to you so you can see them going in. Is that in focus? Yeah. Look at them all. Virgin sacrifice right here. Don't say, I don't give you what you want. That is hundreds, if not thousands, of virgins going into the morgue. Yeah, they're all gone. Done. I will finish labeling these, but that's really boring, so I'll stop now. And yeah, hope you enjoyed that stream. Now you know what I mean when I say that I'm collecting virgins. It is now 7:15, so I'm going to go home as soon as possible and have some food ready for my experiment big day on Friday, when I will be taking one male and one female, putting them together, and then having them mate, lay eggs, collecting the eggs, because then I know that all of the eggs are full siblings of each other. They have the same mother and the same father, and from then those will be my related flies. So yeah, that's what I do. Hope you are enjoying the rest of your day today. Wednesday? Yeah, hope you enjoyed the rest of your Wednesday. Have a lovely evening, and you can tell people that you've just watched me sacrifice thousands of virgins, and you will be telling the truth. There is no word of a lie to that statement. So, bye-bye, everyone.